respective home environments, and then mapping fitness QTL. And with this, they actually have been able to identify loci that potentially underlie local adaptation between these two ecotypes. And one particular candidate, and that will be the focus of this talk, is this exciting and interesting frame shift mutation in a CBF gene in the Italian ecotype. So with transgenic experiments, we were able to confirm that this reduces freezing tolerance in Italy, and based on the condition within QTL, it indicates that it's actually adaptive. So really interesting to think about having a frame shift mutation being adaptive in a warm southern climate, especially because this gene is known to do extensive research, mostly with my clinicians lab at MSU, that CBF genes are central players in conferring freezing tolerance in plants. So just to give you sort of a brief summary of what they do, when plants experience cold temperatures, they upregulate CBF genes. These genes are activators of a number of downstream genes and ultimately confers freezing tolerance. So what I'm going to be talking about today is 3 CBF genes. It's a Paralogous family of transcription factors located in the tandem array on chromosome 4. And so I just pointed out, just to run it right, here's the frame shift mutation in CBF2 that appears to be locally adapted in Italy. And so this sets up a really interesting situation where we can ask, so what is this actually doing at a species-wide level? And so what we're asking is, in this current study, is the variation that we see in CBF genes at a species-wide level, is it associated with climate? And do we see evidence for a hard sweep of one particular allele? Or do we see the evolution of parallel evolution involving multiple alleles of independent origin? And in particular, we ask, do we see the frequency of frame shift and premature stop codons? Are they associated with climate? So to do this, we obtained sequence data, coding regions for CBFs 1, 2, and 3, and 477 wild accessions of Arabidopsis from across its native range. Additionally, we characterize the local environment using 160 climatic variables to describe temperature and precipitation throughout the year. So, first thing we wanted to look at is what is the relationship between climate and polymorphism in CBF genes. And so I'm going to sort of spare everyone the details of this analysis. I'd be really happy to talk to anybody after this. But essentially what we do is we look at the relationship between individual climate variables and polymorphisms in CBF genes using a statistical tool called redundancy analysis. And then in order to reduce the dimensionality of this 116 climatic variables, we performed a hierarchical clustering analysis where we clustered climate variables based on their pairwise correlations. So 4 distinct clusters of climate variables emerged. These are summer precipitation, summer temperatures, winter precipitation, and winter temperatures. And based on the scores that the individual variables received through the redundancy analysis, we found that winter temperature variables explain significantly more of the polymorphisms in CBF genes than other clusters of closely related climate variables. This is consistent with the hypothesis that winter temperatures are driving the evolution of CBF genes, and logical given the known role that this gene family plays in conferring freezing tolerance. So to sort of get a visual of how this gene is evolving across climate, here's a haplotype network. These are haplotypes with greater than 1% frequency in the samples that we looked at. And so the haplotypes are sized by the number of individuals having it and colored according to the climate that they experience. So what you can sort of see is that accessions coming from colder climates tend to sort of cluster together, have a similar CBF sequence, whereas accessions from warmer climates show much more divergence. And so this sort of indicates that as Arabidopsis thaliana populations are moving into warmer and warmer climates, we see independent evolutionary trajectories of CBF genes. So if you remember, there was a frame shift mutation in Italy that looks locally adaptive. Here it is. And so here are the 3 CBF genes, and these are the independent and multiple additional frame shift and premature stop codons that we found in CBF genes that we looked at in this sample. And so what we can see immediately is that there's multiple independent frame shift and premature stop codons, and there are no other accessions with this same frame shift that appears to be locally adapted in Italy. And this is consistent with this sort of parallel evolution. But we wanted to look at, so what is this relationship? What is the relationship between these variables and geography and climate? So here's a map of the accessions that we looked at in this study, and the map is colored according to the minimum temperature or coldest month. And so what sort of comes out is that these frame shift and premature stop codons are generally located in warmer environments, and they appear individual and unique alleles appear restricted to warm clusters or warm patches across this habitat. This may indicate that the flow of such alleles is restricted by places where it's colder, and maybe maladaptive and have a loss of function mutation in one case. We also found that the relationship is significant, so it doesn't just look it on a map. There's actually a statistically significant relationship between the frequency of having a frame shift or premature stop codon in one of your CBF genes and being from a location with a warmer minimum winter temperature. And so here, this plot is more just a visual. We ended sessions by what the minimum temperature is and looked at the percent of individuals within each band having one of these loss of function mutations, and the logistical regression gives us the statistical significance associated with climate. Oh, and here's just to indicate for you guys, this is the minimum temperature experience in Italy, where this kind of allele appears to be adapted. So we also wanted to sort of get a sense for the actual pressure of selection on this gene, whether there's an association with climate. So using these same climate bands that we use to look at the percent of individuals with frame shift and premature stop codon alleles, we looked at performing the McDonald-Kreitman test using Arabidopsis lyrata as our outgroup. And essentially what we found is that for accessions experiencing minimum winter temperatures from -5 to 5 degrees Celsius, they have an excess of non-synonymous polymorphism. So this sort of indicates that, at least at a local level, there is selection for non-synonymous polymorphisms coinciding with this increase that we see in the frequency of frame shift and premature stop codons. Something that really struck me, and at first I was like really confused about this, is, I don't understand why I don't see this, why is there a decrease in this trend towards increased frame shift and premature stop codons in this above 5 degrees Celsius. Additionally, we did not find that accessions above 5 degrees Celsius showed excess of non-synonymous polymorphisms. However, if you go into the literature, since this is such a well-studied gene, you can actually look at studies showing expression patterns as a function of temperature. And essentially, CBF genes aren't expressed above 5 degrees Celsius. So for all the experimental work that's been done on CBF genes, basically if you want to induce CBF expression experimentally, you have to use temperatures below 5 degrees. So accessions that experience minimum temperatures that aren't below 5 might actually be experiencing reduced selection pressure for loss of function because CBF genes basically just aren't expressed. So to conclude, I think this work indicates that winter temperatures are driving the evolution of loss of function of CBF genes in Arabidopsis, and that this work shows that this is occurring through parallel molecular evolution. And I think that this supports the hypothesis that there's some sort of cost to freezing tolerance that's avoided by knocking out CBF genes. And so this work has got me thinking about a lot of things. And so one of the things I've been thinking about is how loss of function mutations have effectively a high mutation rate. So the mutation rate generating loss of function mutations is much higher than some sort of gain of function mutation. So I think that the effect that this increased mutation rate has on the processes underlying evolution might be really important. So parallel loss of function may be an important mechanism underlying local adaptation, especially when climates are changing rapidly, when there's limited gene flow, and when selection is patchy across landscapes. Another thing I've been thinking about is how this has sort of affected the way that I interpret genome-wide scans for selection. So most of the methods used to scan for selection across genomes would probably fail to detect loci that are adaptive if they're evolving in parallel. Just something that I'm thinking about. So some future work that I'm interested in doing is trying to get at, so there's this cost of freezing tolerance, but we don't really understand like what is the actual phenotypic cost, what is the ecological mechanism driving this cost of freezing tolerance. So there's some sort of penalty associated with having freezing tolerance when it's unnecessary. So trying to keep apart why that is would be interesting. Something else, sort of a side thing, so looking at parallel adaptation and processes underlying adaptation to a different climatic gradient, this one being drought frequency. So I've been using satellite data to calculate the frequency of drought across landscapes, And so I think it'd be interesting to look at how molecular processes underlying adaptation of that gradient as well. So with that, I'd like to thank all the co-authors, colleagues, and thank you for your time, and I'll take any